Welcome. So you probably noticed that the title of this presentation, this video, is a little bit different than what's in Carmen. I've decided to not even try to dive into Bayesian inference in this single video. I think that's going to be way too much to try to cover within an hour or so. So we'll hold off on that until next time. Last time we worked our way up through the basics of the Hegelian logic that leads to the inference of phylogenies. And basically what we're doing is we're looking for shared derived characters along the way. Now, what I did was basically talk about doing this by hand, and in simple cases you can do it by hand, but the techniques involved, including the outgroup analysis, outgroup comparison in order to hypothesize which character states are plesiomorphic and which are apomorphic, can be translated into an algorithm such that you can do the work on a computer, which turns out to be absolutely necessary once you get a data set of any size and any complexity. Trying to do all that in your head, I can tell you from personal experience, is absolutely impossible. And so that's why the term that had been used for phonetics, that is numerical taxonomy, kind of was no longer actually unique to phonetics, because the phylogeneticists were as deep into computer algorithms in order to estimate phylogeny as any phoneticist had done for their own work. So let's start out here. Let me grab my pointer. There we go. Well, it's just a little jargon that I think falls a little bit too easily off of my tongue, and perhaps you're not quite as familiar or comfortable with the terms, or have them so deeply ingrained that you don't even think about them. So let's specify here a little bit. So when I talk about daughter species, I'm talking about a situation in which you might have an ancestral species that split into two, and the two species that result from that split, from that speciation event, are daughter species. Don't ask me why they've chosen to add gender to the equation, but that's the way it is. And just as another example of a gender-based terminology, we have the term sister species. And think back to that ancestral species that's splitting into two daughter species. So they're daughter species from the point of view of the ancestral species. From the point of view of either of those daughters, then the opposite one is its sister. So talk about sister species, talk about sister groups. If that split then led to on each side had more speciation events following on later. And so basically we're talking about the two halves of a speciation event. And then there are a bunch of terms that are used to describe these tree-like structures, these hierarchical renditions of the data and the relationships that we're seeing. So there's lots of names. This is not a comprehensive listing of it, but these are some of the familiar ones, or common ones that you will run across. First, we have a phenogram. So a phenogram is going to be anything that is produced in a hierarchical clustering technique that is phonetic in nature. So your neighbor joining trees, your UPGMA trees that you did last time in lab, are examples of phenograms. A cladogram is, well, it can be used in a more general sense, it can be used in a more specific sense. A cladogram is a phylogenetic tree, a branching diagram showing the relationships, the branching pattern evolution, and the phylogenetic relationships among the taxa. So it's basically the branching diagram. If you come across the term phylogram, that's the last one here, that's usually telling you that this is a form of a cladogram, but one in which the length of the branches, so from one node to the next, or from one node to the O2U at the end of that node, that distance is meaningful in some way. Usually it's the amount of change in the characters that are used to infer the branching pattern. Now, I didn't put this one down, maybe I should have. The amount of change that has taken place along a branch, if you assume some form of a clock-like mechanism for evolutionary change, either a molecular clock, or you could, in theory, do this with morphological characters. But you've certainly seen it with molecular clocks. Sometimes that the length of those branches represents time. And so in those cases, you could call that a chronogram. For me, I tend to use cladogram or phylogenetic tree in a kind of a general sense. And then by inspecting it, you can make some inferences about what is being represented in the way the figure is drawn, and then apply this kind of terminology if you want. So we talked about the basics of the Hennigian logic, the way by which Hennig thought it was the logically rigorous way in order to infer common ancestry, to infer the pattern of branching through evolution. There are some consequences of this. Some of these are simply methodological imperatives or rules, and the real world might approximate them more or less closely, but others are really important. One of the ones that's not so important is this first point, but this is one that generated a lot of pushback on the part of the more traditional taxonomists at the time, and that is Hennig declared, if you will, that if we have an ancestral species that's undergoing a speciation event, then once that ancestral species splits into two, so now we have daughter species, for him, that ancestral species ceased to exist at that point. And now we had two new daughter species that had arisen from that single ancestral species. This got a lot of pushback because if you think back to our modes of speciation, sometimes it's thought that pretty well established that species can arise by small peripheral populations over being separated from a larger, more widespread population of species. So we can think of this ancestral species as the original widespread population, and then this small little bubble comes off the side, and for one reason or another, we end up recognizing this as a distinct species. In those cases, that ancestral species, if you look at its characteristics, it probably hasn't changed one whit from what it was before the speciation event. And so people thought it was just crazy to say, well, the ancestral species ceased to exist, went extinct, if you will, because of the origin of this small side population as a separate species. Similarly, the mandate that speciation occurs dichotomously is obviously not true. This is simply a logical, a more clean, logical way to approach the question of inferring common ancestry. But in the real world, it's going to be messier. We can have things splitting off simultaneously, as with a large vicariant event that's like the desiccation in the American tropics, with the retraction of the forest into these refugia around the edges of Amazonia. That probably took place more or less simultaneously, so speciation was taking place more or less simultaneously. We just didn't have this 2 by 2 by 2 sort of breakdown in species. But I think that's more of a methodological point, and even though it did cause a lot of consternation amongst the more traditional folks, it's really not criticism of it is really not fatal to Hennigian phylogenetics in any way. When we get around, we haven't really talked about classifications in the sense that I've been talking about construction of trees, but then the tree that you come up with, the cladogram, the phylogram that you come up with, must then be translated into a classification, and I hope to get to that in a later video. But according to Hennig, according to his principles, all the taxa that you end up recognizing in your formal classification that's derived from your cladogram, all the taxa that are formally recognized must, and I put this in boldface, all caps, underline, I don't know what I guess I should have done italics as well and put it in red. They must be monophyletic. And in this sense, I mean monophyletic in the strict phylogenetic cladistic sense, holophily again, if you will. So that is a mandate of phylogenetics. Paraphyletic taxa, although they were very common in older, more traditional classifications, are in a way an expression of ignorance. And there were lots of arguments about what is the most efficient way to store information in a classification. And for my money, the conclusion was we need a hierarchical classification in which all the groups are strictly monophyletic. Now, some will take the next logical step and say not only must all the taxa that we recognize in our formal Linnaean classification, our ranked classification, not only must all of those named taxa be monophyletic, but all of the monophyletic taxa that are in your cladogram must be put in the classification with names, with ranks. This mandate has largely been ignored because it necessarily would lead to a proliferation in the number of ranks you need in a formal classification with the number of names that you're going to apply to all of those monophyletic taxa. In fact, most monophyletic groups that you see represented in a cladogram, especially if you're talking about something with lots of OTUs, with lots of species in it, most groups are not going to be formally recognized at all. And many of those groups are the confidence with which we recognize them, video next week, preview, might not be the greatest thing in the world. So if you start recognizing them and then new evidence comes in and these things that you didn't have a lot of confidence in that branching pattern before now get changed, well, then your formal classification has to change. You just create chaos, especially you get competing classifications that people disagree in details there. If you've gone this route of the really hard-boiled, hardcore phylogeneticists of demanding that every monophyletic group in a cladogram be formally recognized as a taxon, then for me, this is craziness. But it exists. So, not so much jargon or words here, but some drawing conventions I want to make sure that I cover here. So here we have a cladogram. And we notice, what do we notice on here? You can see the branching pattern going through here. And on the end, see oryza, so that's rice, and I don't know what all these plants are, festuca, so that's a grass. So we got a plant classification here with some outgroups and then this in-group here. All of these numbers and dots and dashes and open circles and closed circles here are these authors' attempts to illustrate for you not only the branching pattern and the relationship amongst the OTUs, but also to give you an idea of the level of support for the branching pattern. And so what they're illustrating here, each of those dots represents an apomorphic character state on this tree. So we can see, let's pick one at random. This group here from Brisa all the way down to Poa lenigera. So this group, arising from this branch here, is supported by 4 synapomorphic characters. Whereas, whoops, I lost my pointer. Here we go again. Whereas, where's another one here? This one here, leading to these 3 genera, Catabrosa, Puccinella, and Sclerocloa. Puccinella, that's nice. Is supported by 5 synapomorphic characters. So a little more evidence there. Moreover, they're telling you what the evidence is. The numbers here, 11, 13, what is that, 29 and 51, will refer you back to a table in the paper where they tell you what those characters are. They've done that's not too uncommon. Sometimes people don't use circles, but just kind of cross hatches here to represent the same thing. These authors have done something a little bit more elaborate, and that is that they're illustrating with an open circle a character that represents a reversal. So this is character 11 here, is a loss, and I'll bet if we go back through here, here's an 11 loss over here. So 11 may have been a character that was very basal here. I'm not seeing it anywhere else at the moment, but in this case they're representing with an open circle a loss. And then the dashes underneath, by their convention, they're telling us that these are homoplasias characters. So not a reversal, but perhaps a convergence or parallelism, which in fact are often operationally difficult, if not impossible, to distinguish. I didn't really look at the caption that carefully. These losses may be just what they say, a loss, that something is not there, an absence, and not necessarily a reversal. But you can see that oftentimes homoplasias characters are included here. In this case, it's saying that, what is that, character 69? It's saying that somewhere else on this tree, this same character state evolved. And so in this context, it's a synapomorphic character. And wherever else it shows up, it may also serve to unite OTUs into monophyletics groups. So a lot of that is non-standard. I mean, to have crosshatches and numbers which refer you back to a list of characters, that's kind of normal. But the rest of this stuff is non-standard, and so you need to read the captions in order to make sure that you understand exactly what the authors are trying to represent. They don't make any assumptions there. Let's see, what else do I have here? Do branch lengths mean anything? Well, let's take a look at this. And in this case, we see that, for example, this branch is much longer than the one it subtends here. And so in this case, it does mean something, that the branch length is proportional to the number of synapomorphies on that. In other words, it's proportional to the amount of evolution that the authors are inferring has taken place along this branch. I should also, let's put it this way. It's very easy to look at something like this and interpret these branches as ancestral species. That's a dangerous route to take. Strictly speaking, what they are representing is the distribution of shared derived character states among the OTUs in the study. You can see how you can get to the inference that they're an ancestral species down here, but there's really there really are no OTUs that are being scored with these characters down here. That would be a logical inference, but it's not exactly what's being represented here. Does the order of OTUs mean anything on this tree? Well, in one sense, it does. That Poa palustris and Poa lenigera down here are right next to one another, and then joined at this node, tells us that these are sister species. So in that sense, it does. But does it make any difference that they listed Poa palustris above Poa lenigera? Or could they have put lenigera above palustris here? That aspect of the order is there's no meaning to it whatsoever. What I like to think of, this is the way I was taught it in an ecology course, is that you can think of trees like this as similar to the kind of mobiles that you hang above a child's crib. And you know how they spin along the nodes? So you can imagine, turn this 90 degrees, what is that, clockwise, and then hanging above the baby's crib, and these things spinning around on the nodes here. The topology, the relative position of everybody stays the same, even if I spin this one 180 degrees, spin this one around any of those nodes. So in that sense, the order of the names of the OTUs does not signify anything. What's important here is the topology, the shape of the tree. Finally, where is it? Here's one. What is the significance of these polytomies? So the Hennigian principles, you're supposed to be dividing. Every time a speciation event occurs, you end up with two daughter species. So it's dichotomous branching, from one branch going to two branches. So where do we have? 1 to 2, 1 to 2, 1 to 2. But occasionally, like down here, we have a polytomy, that is many branches coming off of a single node. In this specific case, this is a tritomy with 3 branches coming off that one node. What does that mean? Well, it can mean a couple of different things. It can mean that there is no evidence, there are no synapomorphies that unite any of these 3 branches, any one of these 3 branches to any one of the others. So no synapomorphies uniting this branch with this branch with this branch, or this branch with this branch. So in the absence of data, we'll be right out there with our statement of ignorance that we don't know how they're interrelated. This may also reflect that the underlying data conflict with one another, and I can't choose amongst them. Maybe there's one synapomorphy uniting this one and this one synapomorphy uniting the first and the third, and one synapomorphy uniting the second and the third. How do I choose amongst them? I can't. Therefore, I'll represent this as a tritomy, or a polytomy in this case. So basically, it's a statement of ignorance, either because of the lack of data or because the data are ambiguous and do not effectively resolve the relationships. By resolution, I mean breaking it down into dichotomous branching. So a fully resolved tree will have no polytomies here. This polytomy here is a little bit special because this is the outgroups. And so this is where the outgroups arise. So really, we're concerned with the in-group here, and you're always going to have a polytomy here at the base, or that's what the analysis will do. Sometimes people, as you'll see in the next slide, will draw a root coming out here. So synapomorphy is indicated. We have to examine the tree to make sure we know if there is any meaning to the branch lengths. Not so much the order of the OTUs, but how they're linked together. That's what I mean by the topology of the tree. That's what's critical. And each of these nodes you can spin around and present them in any shape you want. And then the polytomies. Let's take a look at the next one here. Rooted versus unrooted trees. I suppose our analogy with plants, it might be getting in the way here a little bit. But basically, another way people call these is directed versus undirected graphs. So on the left here, we have a rooted tree. Actually, both of these illustrations are expressing almost exactly the same thing, with one exception. So here we have a bunch of, they're calling them leaves. I've been referring to these as the OTUs. So we have 1, 2, 3, 4, 5 OTUs. And over here we have 5 OTUs. Then they've also numbered the internal nodes here. And when you add a root to it via outgroup analysis, that then specifies the pattern of relationships amongst the species with. Again, this is more an inference rather than actually what the data are showing here. That time is implied, but it doesn't mean that the time is proceeding monotonically. Again, in this one, we can see that branch lengths appear to have some meaning here. So say this is representing exactly the same thing, except we don't have a root. If you look at here, we have 2 and 3 grouped together at node 6. 2 and 3 grouped together at node 6. That grouped together with 1 at node 7. That grouped together with 1 at node 7. So this is exactly the same topology. It simply has not been rooted. And one of the interesting things about the algorithms that are used, we're going to start to get in here a little bit, and you'll be getting into in lab, the algorithms actually start out by calculating the unrooted tree and then add the root to it. So the initial calculations are exactly the same. And where you place the root, in this case, the root has been placed, they call it node 9, but really it's between 7 and 8. 7 and 8. So right here is where that root is going to be applied. And so then that would imply that 5 and 4 form a monophyletic group, and bingo, they do. And 1, 2, and 3 form a monophyletic group. Bingo, they do. Also notice you can draw this tree facing any direction that you want. This one's pointing, having the OTUs at the bottom. I've shown you trees with the OTUs at the top, at the right. Maybe left-handers have the OTUs on the left, or if you're trying to compare two trees side by side, people would do that. So that has no significance either. This is also a cladogram, but in this case, it's represented not with those rectangular, boxy sort of nodes and branches, but as spokes coming off a circle. So first question is there a root? And if you go find the root, you go right down to the middle, and here's the root of this tree. You can also express this, and in something like Mega, it's just with a click of a button, you can switch between the different modes of representing these cladograms. So here's a root, and you see we have a basal polytomy. So as I said, these are probably the outgroups here, or at least this one's the outgroup coming off. And then the topology is represented around the outside. In this case, we can see that we have different, let's see, do we? No, we do not have different branch lengths, although it's got a tree scale, so we must. Yeah, this is a long branch. This is a long branch. I was just looking here, and these all seem to be close to the same, but the fact that they're giving you a scale here, and the units are probably in the caption, you know, nucleotide changes per 1,000 bases, 1,000 sites in the sequence or something like that. It'll give you an idea of what the branch lengths are supposed to imply. Fundamentally, it's implying the amount of evolution that has taken place along those branches. People like to use these circular diagrams when you get a large number of OTUs. I put this one on because it's very simple, but you can imagine if there were 500, and I've seen some like that, then this is going to be very dense going around here, and they always become very difficult to interpret. In fact, it's kind of a standing joke when you go to meetings and see presentations with well, any sort of tree structure. If the OTUs are abundant enough, then the first thing that the presenter always says is, I know you can't read this, but let me focus in on for you. Cardinal sin of giving a presentation, never apologize. Get it right. Now I know you can, no, sorry. So let's move on. The simplest form of a cladogram is what's called a 3-taxon statement. So by that I mean, given 3 OTUs, say species A, species B, and species C, The cladogram is going to represent the pattern of relationships among these. Remember, this is the minimal number. You always have to have 3 in order to have a meaningful cladogram, because any pair is going to have a common ancestor somewhere along the line. And you're just leaving out lots of intervening material. So given that I have 3 OTUs, how many different ways could they be related? Well, it could be that species A and species B are more closely related to each other than either is to C. That is, A and B share a more recent common ancestor, represented by that node and I guess by that branch there, than they do with C. So that's one. It could be that it's not A and B that are most closely related, but A and C that are most closely related. Or it is B and C that are the most closely related pair, and A is on the outside. This really is the grand total of all possible rooted trees expressing the relationship amongst 3 taxa A, B, and C. As an exercise, I challenge you to convert these into unrooted trees and see what the unrooted tree looks like. You might be surprised. So for 3 OTUs, there are 3 possible rooted trees. Which one is right? Well, we have to examine our data in order to tell us which one is the correct or the best relationship. So 3 trees for 3 OTUs. How about 4 trees? Well, one way to calculate that is to take your 3 trees for 3 taxa and now add a new taxon D. So I could take D and say, oh, it's closest to A and put it right there. Or maybe it's closest to B, put it right there. Or maybe it's closest to C and put it right there. Or maybe it's closest to the combination of A and B and have it arise here. Or maybe it's coming off down here. So I have 1, 2, 3, 4, 5 different possibilities to put D on this tree. 5 over here and 5 over here. And so you can see that the number of possible rooted trees for 4 taxa is 15. So for 3 taxa it's 3, 4 taxa it's 15. We can generalize this with an equation where n sub r that's the number of rooted trees, and the little letter n stands for the number of OTUs in your analysis. So what is that? That's 2n minus 3 factorial times 2 to the n. Something's missing there in my formula. N minus 2 squared. I've actually done the calculations here for up to 10 taxa. So as I illustrated for you, for 3 OTUs, there's 3 rooted trees. 4 OTUs, there's 15 rooted trees. 5 OTUs, there's 105 rooted trees. You can see this is not increasing linearly. 6, 945, 7, 8, 9. When you have 10 taxa, there are 34,459,425 different ways to express the relationships among them. In our current project, we have approximately, I think it's 200, a little over 200 taxa in our analysis. This number is probably larger than the number of electrons in the universe. It's a huge number for that many OTUs. This has a couple of implications because some of the methods for trying to find the correct tree given the data that you're using involves evaluating each and every one of those trees. Obviously, if you have a large number of taxa, then that's going to be probably physically impossible, and certainly it's going to take way too much time, even with your fastest computer processor, for you to explicitly evaluate your data against every possible relationship there. So we have to use more approximate methods in order to do this. And that leads us to the topic of optimality criteria. given that there are many ways, many different trees that you might come up with, there are many different trees that can express the relationships amongst them. And once you start using approximate methods to find the correct tree, then you're going to find a lot of trees that are, can I say this, equally correct, look essentially similar, are differing only in details. How do I choose amongst them? Which one is the best tree? And there are different ways there are different criteria by which to measure best. And I'm going to talk about two of them in the remaining time on this video. The first one is our old friend from last time, the parsimony argument. So in this, for this criterion, if you're using parsimony, you're saying basically the best tree is the one that is going to require the least number of ad hoc hypotheses to explain the observed data. So let me give you an illustration of what that means. So I've gone back to my three taxon statement because that's the simplest way to represent this. And on the first one, these little dots here are supposed to represent the distribution of derived characters. So I've got, 4 characters here. And we're assuming that the same distribution of characters is across all 3 trees. So this is A has these 3 derived characters, B has these 4, and C has this 1. And I matched the color to say that A and B share this derived character, share this derived character, and share this derived character. But up here, this red pair, that derived character is shared by B and C. So we have conflicting data. Some of them tell us that A and B is most closely related. And the other one, the red one there, tells us that B and C are most closely related. Now down the bottom on the cladogram, what I've basically done is mapped those derived character states onto the cladogram. Or another way of saying that is that I've mapped where that character changed from the plesiomorphic character state to the derived character state. So for this character here, this is telling us that C has the plesiomorphic character state. So I'm only showing where the derived characters appear. So in this case, are A and B the most closely related? Well, we have 3 characters that say yes. In this one, well, let's do it this way. We have 3 characters that say yes, and I've mapped the distribution of character states on the other 2 possibilities for these 3 species. Now let's count them up. This tree, this cladogram, requires 5 steps. An apomorphic character change here, an apomorphic character change here, a change to an apomorphic character here, and then B and C in this scenario must have, looks like I lost a red dot right there. Anyway, must have evolved this apomorphic character state independently of one another. So I'm hypothesizing 5 character changes, or typically we use commonly we use the term step. 5 steps here, 3, and then 2 more here. In this pattern of relationships, I have to hypothesize 8 changes in character states, or 8 steps. So 3 leading to A, but now because A and B are separated, B has to evolve these on its own as well. And then C and B independently evolving the red apomorphic character state. So that's 8 steps. And you can do the math here. It's easy. For the hypothesis that B and C is most closely related, we have to hypothesize 7 character state changes here. Now, astute listeners will say, well, maybe some of these evolved earlier and then were lost later on, or reverted back to the plesiomorphic character state. That's true, but if you do that, you'll find that the number of character states ends up being exactly the same as in this in this scenario here, 5, 8, and 7, no matter how you do that. So the principle of parsimony, or if you're using parsimony as your optimality criterion, which of these three trees is best? Which number of ways to say it? Which of these three possibilities best explains the distribution of derived character states in the trees? Well, according to a parsimony criterion, it's the one with the least number of steps, 5 here, which makes intuitive sense. There are 3 characters that unite A and B, and only 1 character that unites B and C. So 3 to 1, we have a democracy here. Outvoted. This is our best tree. So, use of parsimony was probably the first explicit criterion that people applied when trying to choose between different representations of the data in the form of cladograms. There were some others, but parsimony gained a lot of adherence because I hope that I explain that in such a way that it makes common sense to you. Also, at the time that these developments were being made, theoretically, the data that were available were largely anatomical data. I mean, to be sure, DNA sequencing had been invented and protein sequencing had been invented, so you could in theory get sequence data, but methodologically it wasn't until the advent of PCRs that was done, and Sanger sequencing, that was done in any large economic scale. So people were dealing with morphological characters, and so maybe you're talking about the appearance of a seta on the hind coxa of an insect, or 2 setae. So from 1 to 2, we have to evolve. We're talking about an increase in the number of setae. Maybe it wasn't 1 seta to 2 setae. Maybe it was no setae to 50 million setae on the body. You know, you have a cetose body, you don't have a cetose body. We're talking about the ways in which you encode the characters. It's a topic for another time, but basically, the point I'm trying to get to is that with morphological characters, there's usually no real basis to say that a change from 1 seta to 2 is any different fundamentally than the change from a wing with a pointed tip and a wing with a rounded tip. How do you equate those two? They are 2 changes in a sense, but what's going on underneath the covers in terms of developmental processes is largely still totally unknown. And so since there was no way to say, well, this is a big change and this is a little change, people usually just dealt with them in terms of the number of evolutionary steps. So there was some kind of implication that down deep, 1 step in the change of the number of setae on a coxa was equivalent to a step in the change of the shape of the apex of a wing. Whether that's true or not, you could argue. Sometimes you can break those characters down and say, well, actually, to get the wing in that shape, it takes 2 or 3 changes. And you can specify what they might be. And there you're breaking down a complex character into its more atomic subunits. But at the end of the day, what you're doing is counting steps and making the assumption that those steps are in some way equivalent. And that may well not be true. It's almost certainly not true, but without any explicit way of going to an alternative, that's what people have opted for. I actually didn't put that on a list of problems with parsimony, but it probably should be there. Another problem with parsimony analyses is that it turns out that when you do it, it's not unusual. In fact, it's largely the rule that there are many equally parsimonious solutions. So instead of just choosing between options that have 5 steps versus 7 or 8, and the answer is clear, you might have, 25,000 trees of 5 steps. Which tree, they're all different in their topology, which tree is the best? Don't know. They're all equally parsimonious. I have no way, if that's my criterion for choosing best, then I have no way to select amongst them. So that turns out to be very common, and also that evolution of morphological characters turns out to be much more homoplazious than one would ever expect. What we look at and see as structurally the same thing turns out on the basis of independent analyses using independent data, and in this case it's usually molecular data, that the morphology can be actively misleading. Anyway, many equally parsimonious solutions could exist, and those are usually expressed in those polytomies, that are statements of ignorance about what the right answer is for resolving that polytomy into a series of dichotomous relationships. Then there's a phenomenon called long branch attraction that really came to the fore as the molecular data started to overtake. And why did molecular data overtake morphological data? After all, what we're often most interested in is the evolution of morphological structures, because that's how animals, insects, I suppose plants, do things, and we're interested in what they do and how they evolve and the selective pressures and all this kind of stuff, and not necessarily in what a particular nucleotide is at a particular place in a particular protein. But the attraction of molecular characters is the sheer number. That you can have many thousands of times more characters, the individual positions in a multiple sequence alignment, to give you signal about what the relationships amongst the taxa are, if you use molecular data as compared to when you do the morphological. To go back to example from my lab, in our 200 OTUs, I didn't count them up, but I think we have somewhere on the order of 150 or so morphological characters. 150 morphological characters for 200 OTUs means that some taxa could well be exactly the same. We don't have enough characters in the morphology alone to fully resolve that. In comparison, I didn't look up the count, but we have several thousand molecular characters. And for the phylogenomic one, we have several thousand genes to distinguish those taxa. And so molecules have taken over just for the sheer volume of data. Now, those data can be problematic, and this last phenomenon is one thing that reared its head here. It's a phenomenon called long branch attraction, and basically what this is if you're looking at a nucleotide sequence, then there's basically, for a single position in an alignment, there's basically only 4 bases that you can have there. You can be A, C, T, or G, right? So it's possible that 2 OTUs at a particular position in a sequence alignment will have exactly the same base there, not because they inherited that from their common ancestor, but because of pure chance, that there was a mutation there, and from the A that it was, for example, it could only go to T, C, or G. There were only 3 possibilities for it to mutate to, and so if it's got a C now, then that happened, and somebody else has a C, that happened totally by chance. So it was first shown theoretically by Joe Felsenstein from the University of Washington that under certain conditions, the parsimony criterion for choosing among trees resulted in anomalous, wrong relationships being inferred. And for a while, that was called the Felsenstein zone. Did you meet those conditions that he posited? And the way it works is that if you have a branch on your tree, and the species at the end of that branch has evolved to be very, very different from all the other species, then it is possible that in the sequence alignment that you're comparing, that it will share what look like similarities with quite distantly related taxa, but that appearance of relationship, the sharing of those characters, is due totally to the chance mutation to the same base at a series of positions. And so you could get a misleading answer if you have taxa in your analysis that are at the end of very long branches. In this illustration, I believe it came from Felsenstein's paper here. Species A here there's been a lot of stuff going along here. So the length of the branch is representing the amount of evolution through time along it. And so it might accumulate differences that now suggest that it's most closely related to C, when in fact it's most closely related to B. So, theoretical problem or a real world problem? Let me give you an example from the insect world. If you're not familiar with this creature, then hopefully by the end of the course you will be. This is the insect order Strepsiptera, or the twisted wing parasites. On the left here, we have an adult male, and these are really bizarre little creatures. This guy is probably about 2 or 3 millimeters long, so not huge. They have these big fan-shaped hind wings. If you look in closely, this picture is too low resolution to see it, but they have these big, giant, raspberry-like eyes, so big ommatidia on the eyes. They have these branched antennae, and then these little things you see here are not legs, but these are actually the reduced forewings of the creature. So they're kind of like club-shaped organs here. On the right here, this is a Polistes, this is a paper wasp, but the strepsipterans are these little black things poking out from underneath the abdominal terga of the paper wasp. This is the male, and in the immature stages of the male, and the entire life, except for the very first instar in both of them, the rest of the life of the female is spent as an internal parasite of other insects. Paper wasps are very common, bees, leafhoppers, things like that, and the silverfish, interestingly enough. So these are either females that are poking out or a male that has pupated and ready to come out. So these are the internal parasitic forms, these are the external forms. The male and that first instar are the only non-parasitic stage, the adult male in Strepsiptera. So the question is, what are Strepsiptera most closely related to? I hesitate to say classical, but the older hypothesis was that Strepsiptera might be, they're not similar to anything. They've changed a tremendous amount. The stereotypical long branch on a phylogenetic tree, they've changed so much as internal parasites and their structure. But the idea was, well, these guys, the males, the only flying stage, are relying upon the hind pair of wings in order to propel their flight. That's called posteromotorism. A big name for meaning that they're basically using their hind wings. Whereas this Polistes, although it has hind wings, they're small, and it's using the forewings to propel itself, to generate the majority of aerodynamic forces for flight. What other insect group has posteromotorism, uses hind wings? Well, the most obvious example are the beetles. that they rely on their hind wings, and then they have the sclerotized elytra that are more than anything else just protective organs for when the insect is walking around, tucks the hind wings underneath there. Another group, Dermaptera might be, but these are holometabolists. They have pupae. So amongst the holometabolous insects, this use of the hind wings suggests that maybe beetles are their closest relatives. And in fact, there are some beetles that are, there's some pretty bizarre beetles, and some of them kind of look like male Strepsiptera. And so that was kind of the reigning hypothesis. That is, until somebody, in particular Mike Whiting and Ward Wheeler, at that time from the American Museum of Natural History, tried to do some sequencing these things. Mike is now at Brigham Young University, and Ward is still at the American Museum of Natural History. They published a paper here on the left in, what was it? I can't remember if it was Science or Nature. It was one of the big boys. In the early days of sequencing, they were looking at 18S ribosomal DNA. So it was still in such early days that they weren't doing dozens or hundreds or thousands of genes, but just doing one or two. But their analysis suggested that when they analyzed their data from 18S ribosomal DNA, it told them that the Strepsiptera should be grouped not with the Coleoptera, but with the true flies. Well, true flies, the Diptera, do rely upon a single pair of wings for flight. Unfortunately, it's not the hind wings, it's the forewings. But in the same way that the Strepsiptera have their anterior wings reduced to these little club-like structures here, they're pointing to them H here. They're calling them H because they hypothesized that they were homologous with the halteres of a dipteran, which are also club-shaped organs. The fact that in the true flies, it's the forewings that are normal and the hind wings that are reduced to the halteres, and in Strepsiptera it's just the opposite. They took a leap of faith, and I guess this is the kind of thing that in those days would get you published in Science or Nature. Suggested that there was some sort of homeotic mutation such that the expression was reversed in these guys. An interesting suggestion. Got lots of press, lots of interest, lots of head scratching, because to be honest, the Strepsiptera don't look a hell of a lot like beetles. So here was an intriguing, revolutionary hypothesis about what's going on based upon the sequencing of their molecular data. Well, that was followed up by an article that said, well, you know, actually, what we have here is an example of long branch attraction, that the Strepsiptera have evolved to be so differently, and their sequence, their DNA sequence, their 18S DNA sequence has evolved to be so different from their closest relatives that that branch was attracted, as it were, to another branch farther out in the phylogenetic tree, in this case, the true flies. And that, in fact, when you analyze the data, well, no, this is not a reanalysis. This is the straight up data. So here's the Strepsiptera up here being attracted, being grouped together with the true flies. What do we have here? Aedes, mosquito, and Drosophila here. And the beetles are somewhere down in here. They are Meloids and Tenebrio here. It was some of the Meloid beetles that similarity between male streps and was pointed to, that perhaps they're Meloids. That's what my old advisor suggested at one point. Perhaps they're the sister group of beetles. And so they came to the conclusion that the DNA data were actively misleading Whiting and Wheeler, into an incorrect pattern of relationships. And then that in turn led to the totally unnecessary hypothesis, speculation of some sort of homeotic mutation that switched. And so now mesothoracic genes were being expressed in the metathorax, and metathoracic genes were being expressed in the mesothorax. Just to, at the point that this was published, it was basically looked at, should we analyze it this way or should we analyze it that way? Well, recently for me, maybe 5 years ago or so, a group in Germany compared not just one gene, but thousands of genes, analyzed them all simultaneously. And we ended up back where we started, that Strepsiptera are the sister group of Coleoptera. They're the ones they're most closely related to. So long branch attraction apparently is a real thing that can be of significance. Sorry. Where do we go next? Okay, so we have parsimony as one criterion for choosing among trees. Another one is one that is, to be honest with you, one of the most popular ones today, is a criterion known as maximum likelihood. So in vernacular, it's probably common for people to use terms like likelihood and probability more or less synonymously, but in fact they mean two different things. So what is likelihood? So I have sort of a symbolic representation of it here that I want to give you, because we're going to delve into this a little more deeply when we talk about Bayesian analyses. But if we represent the hypothesis with the letter H, so in our case, a hypothesis would be a tree, a cladogram, and D as the data, so our DNA sequences, for example, because this is, you can use maximum likelihood analyses with morphological data, but it's more straightforward to use it with molecular data. Then what this is the symbolic representation, this is the wordy representation. The likelihood of your hypothesis, that cladogram you have, given the data that you have, is equal to some constant. We don't know what that constant is, but some constant times the probability of getting those data if the hypothesis is true. The probability of the data given the hypothesis is proportional to the likelihood of the hypothesis given the data. So the best tree amongst all that you might be able to find is the one that's going to have the greatest likelihood. So basically we want to maximize the likelihood. A couple of interesting, or I think they're interesting, peculiar, maybe is another word for it, things about likelihood is, unlike probabilities, if you look at all the probabilities for a certain event, then they have to sum to 1 because that's 100%. It's not true in likelihood. So likelihoods do not need to sum up to 1. And because likelihoods are proportional in some way, that K value here, to the data that you have, that likelihood values are dependent upon those data. So it doesn't really mean anything to compare likelihoods from two analyses that are using two different data sets. So you can't compare across studies, and the actual value of the likelihood is, in a sense, meaningless. It's the comparison within the study that the meaningfulness, the meaning comes through. Likelihood values are always less than 1, and so less than 0, and so therefore they're usually expressed as the negative log of the likelihood. So you end up with a positive number for comparisons. So let's dive into this a little bit. What about that hypothesis? What goes into making the hypothesis of that tree, of that phylogeny, of that cladogram? Well, it turns out there are lots of things that go into there. There's the topology, the pattern in which the OTUs are connected to one another. The branch lengths are another aspect of it. And then finally, well, not finally, but critically, is the question of how does DNA evolve? How can we, if we're looking at the probability of those data, we need to have some sort of model of how DNA evolves. And this is what I want to end up this video on. So we need models that we're going to describe the ways in which, if we're talking about one position in a sequence, how that nucleotide changes over time from its plesiomorphic state to a new state. So we can express that as the probability of that change. There's only 4 possible states to look at. And so there are models that express how you go from one nucleotide to the next. What's the probability of those changes? Now there's a lot of simplifying assumptions. There's a lot of models, first of all. And it's going to seem like I'm giving you a ton of them. But basically I'm going to go from the simplest to the most generalized model. Even so, there's lots of simplifying assumptions that are made in all of these models. One is that the different sites in a DNA sequence evolve independently, such that we can treat them as individual characters, that they're not, there's no autocorrelation. If you have an A at one position, it doesn't influence what's at the next position. Definitely not true, but we need to make that kind of assumption in order to do the analyses, in order to do the in order to do the calculations. We're going to assume that character state changes are what's called time reversible. That is, you can go from A to T, or you can go from T to A, or any other. You go back and forth. There's no prohibited paths. And then finally, we're making the assumption that the sequences we're looking at are in evolutionary equilibrium. That, yeah, you're going to have random mutations going on here, but you don't have any directed change here. We're basically at a stable state where we can look at these and make some interpretations. So with those assumptions, let's take a look at some of these models. The simplest model is known as the Jukes-Cantor model. These are all named after the people who proposed them in the literature. The Jukes-Cantor model has 2 parameters. It's basically only allowing 2 things. It's making an assumption that all 4 bases of a nucleotide, A, T, C, and G, occur at equal frequencies in the sequence that you're studying, and that they have equal mutation rates. That is, to go from an A, the probability of going from an A to a T is equal to the probability of going from an A to a G to going from A to C. Do all 12 possible combinations. The probability of any change to any other change is equal. Definitely not true, but at least at the point in time where this was suggested, this was necessary in order to do the calculations and in order to see how this stuff could work. You can think of it as an approximation to make the calculations easier. You would get to a better model than you would with, let's say, a parsimony analysis. Take a next step up in complexities, the Kimura 2-parameter model, which makes the same assumption as Jukes-Cantor, that the bases occur in equal frequencies. But instead of assuming that any change, and in this diagram here we have, let's see, adenine here, cytosine, thymine, and guanine at the vertices of this square here, and we're talking about the changes from one to the other. So in this case, from adenine to thymine, it's represented by that blue diagonal, and from adenine to guanine by the red vertical line. In the Jukes-Cantor, the probabilities of each of these lines is equal. They're all the same. But the Kimura 2-parameter model allows you to be a little bit more realistic to say that the probability of a transition, that is from adenine to guanine, or from cytosine to thymine, those might occur at one rate, but transversions occur at different rates. All the same transversions are occurring at the same rate, represented by the blue lines here, but the transitions, the red lines, could occur at a different rate. So instead of having one constant rate of change amongst all nucleotides, we have two transitions and transversions. Then we graduate up to the Tamura 3 parameter, which adds the possibility of allowing for GC bias. So we're no longer assuming that the nucleotides occur in equal frequencies in the sequence. Often this is true, especially with things like nuclear protein coding genes, that within a margin of error, the proportion of guanines and cytosines in the sequence is roughly equal to the proportion of adenines and thymines, so AT versus GC. But when you look at the mitochondria, and especially in insects, you have a tremendous GC bias, that those nucleotides do not occur anywhere near the same frequency. So the Tamura 3 parameter model allows you to do that. So it's been used a lot for insect mitochondrial sequences. HKY85, Hasegawa, Kishino, and Yano. What do we have here? Unequal base frequencies. Okay, we like that. Again, transitions and transversions allows you to differ on those, and allows the transitions are usually more common than transversions. I'm just making it a little bit more complicated model here. Tamura, adding one more. I don't think I even want to go through all the difference here, because as you'll see in a moment, that's not the critical element here. Eventually we get to the most general model, called the General Time Reversible Model, or GTR, which basically posits that all holds are off. We could have unequal frequencies of all four bases. You can change from any base to any other at a rate that is different from, you could always change from any base to any other, but the rates of any particular change can be different for all 12 possible changes. So there's a lot more parameters that have to be estimated. So we're going from a very simple model, Jukes Cantor, up to the most generalized, most robust, most realistic, if you will, model here with the GTR model. So we have lots of models of evolution that we're going to use in our maximum likelihood calculation to figure out how, given the data we see in nucleotide sequences amongst a series of OTUs, which hypothesis has, that is the cladogram, has the highest likelihood of occurring given those data. Now, the question you should probably be asking is, I forgot to keep count. What did I have? 6 different DNA models. Which one should you choose when you do an analysis? For that, what people use these days, and perhaps some of you have done this in ecological problems as well, using a statistic called the Akaki Information Criterion, or just AIC, which is a way to try to estimate the relative quality of the statistical models that are underlying your analysis. So the way this works is that, and of course in the real world the way this works is that you press a button on your screen and go away and come back in an hour, a day, a week, who knows, depends on your data. You compute a tree under these different models, and you can specify which ones you want, or you want all possible models. And for that tree, you compute the likelihood. So your getting a maximum likelihood tree under the different models, and a likelihood value pops out. Now this value is comparable because you're using the same data, same sequence data, same OTUs in the data. So in this sense, the likelihoods are comparable. And so what you're going to do is compare them using this little formula here, the AIC, ICACI information criterion, which is equal to 2k minus 2 times the log likelihood. Okay, so what does that mean? K is the number of parameters that you're estimating. So in the the Jukes-Cantor model, we are estimating, we have to come up with an estimate of the probability of change of the nucleotide. It's all the same for all possible nucleotides, but we only have to have one number that we're estimating there. Whereas in the general time reversible model, there are 12 possible changes, and we have to come up with 12 estimates of that. And where do those estimates come from? They come from the frequencies that we see in the data that we have at hand. So 2 times the number of estimated parameters minus 2 times the log likelihood. So basically what this is telling you, let's say that we've done general time reversible model. So this value here is 24 minus 2 times the log likelihood. So that value is going to, depending upon what the log likelihood is, but you're adding, because this is a negative value, right? You're taking 24 and adding some number to it. Whereas this number is going to be smaller if you're recognizing fewer parameters. So basically the bottom line is that adding more parameters in your analysis will lead you to a higher likelihood, but you're going to be penalized because of the number of parameters you come up with. And so you'll get ICACI information criterion values for all the models. So let's say GTR, some number, Jukes-Cantor, some number, and from that sequence of numbers you choose the highest value. So it's kind of combining the inherent ability to get a higher likelihood model with more parameters with the penalty imposed because you're using more, you're estimating more parameters there. And you just take the highest value. Another criterion that is used is very similar to the ICACI information criterion called the Bayesian information criterion, which just makes the penalty a little bit bigger here. Instead of taking 2k, it's the log n times k here. So which one of those is better? That's simply a matter of taste. So all of these things that I've been describing so far, you will see when you do a maximum likelihood analysis in MEGA, that you will be asked, what model of DNA evolution should I apply? Should I being the software. Which one do you want to be applied? And you can choose amongst them. And when you choose amongst them, I'd like you to choose several and observe what happens given the input that you give the program in terms of the assumptions that it's making. One other thing that you will see that you have the ability to play with in MEGA when doing a maximum likelihood calculation concerns an assumption that we've been making that the rates of change of nucleotides at a particular position in the sequence is the same throughout that sequence. So that the same rates of change in nucleotide position 1, or the rates of change of nucleotide position 1, the probabilities of the rates of change of nucleotide position 1, it's getting to be a mouthful if I keep adding qualifiers to it, is the same as at position 557. That may, or usually is not, a valid assumption, especially if you have proteins. For example, if you have proteins that must be inserted into the cell membrane, then the probabilities of change of certain amino acids are going to be different because they have to have the hydrophobic characteristics of the transmembrane domains of those proteins. So it's usually not a valid assumption that the rates of change are constant throughout the entire protein that you're looking at, let alone if you start comparing different proteins. So what can we do about that? Well, you have another parameter you can adjust in Mega, and in any good, robust phylogenetic analysis program, of which there are lots, that can accommodate this. This phenomenon is called among site rate variation. So you can add new parameters to it. Basically, you can tell it how much variation there can be in nucleotide mutation rates. And these are the two factors. So basically, you're going to estimate this variation, the amount of this variation, using what's called a gamma distribution. And also, you can add invariant sites. There's some controversy as to whether adding invariant sites is valid or not. Mega allows you to do it. RaxML, can't remember. We'll talk about RaxML next time. If it doesn't allow you to do it, but definitely the developer of RaxML says don't do it. Anyway. So what is a gamma distribution? Here I'm going to just demonstrate how to dive in the deep end of a pool of mathematics and drown, or at least, well, actually what I'm going to do is I'm just going to point at the pool and say, gee, look how deep it is. So here we have the graph at the top is some sample gamma distributions. The formula for a gamma distribution, formulas for a gamma distribution are down here on the bottom. This is the probability density function for a gamma distribution, and you can see we have an f of x, a function, and we have two basic parameters here, alpha and beta for this function. Let's this puppy down here. This is the gamma part of the gamma distribution, which is defined in this value here. And what this graph is doing is just showing you what this function looks like when you vary the alpha component of it. So alpha of, 50, gives you a graph that looks like the sort of standard normal curve that you would see in a textbook. Looks like. Alpha equals 200, much less intrasite variation. We have we're most in a graph in a curve like this. If this gamma distribution is describing the variance in the variation in substitution frequencies in it, this is going to have much less variation than this guy, or God forbid some of these here where we're getting very low values of alpha. So the greater alpha, the more peaked the curve here, the less variation there is in substitution rates at a position in the tree. When you get in and do these analyses and you say, I want to apply a gamma function to account for intersite substitution variability, then the program will estimate those shape parameters for you such that it matches what it's observing in the data at hand. So you don't need to worry so much about that. Anyway, so maximum likelihood is one of the prime methods that is used to analyze data from a phylogenetic point of view. Note that I haven't talked a bit about outgroup comparison or things like that, because this happens all under the covers by that mode of calculation that I described. At first it calculates the unrooted tree, and then when you specify the outgroup, that determines the root, and all the business about what is hypothesized to be plesiomorphic and apomorphic takes care of itself. Let's see, where do I want to go now? Just the summary now. So my goal here was to give you an introduction to what you'll be seeing in the lab in terms of the parameters that you can play with in a maximum likelihood estimation of phylogeny. So that was the point of the details. The biggest thing is to introduce these two optimality criteria. So the first point I want to make here in the summary is that the number of possible trees to represent relationships among taxa increases rapidly with the number of OTUs, such that you don't have to go very far before you have outstripped your capacity to actually look at and compare all the possible patterns of relationship. These optimality criteria give us an explicit operational criterion by which to say this is the best tree, or the best tree is at least this. The parsimony criterion is basically the shortest tree is the best tree, which leads to some problems. It makes a lot of sense, and it made a lot of logical sense to me, and I was a big parsimony adherent until we started getting to molecular data, when it was just giving us demonstrable nonsense. But in terms of morphological characters, it still makes a lot of sense. But you often end up with many equally parsimonious trees, such that when we can analyze our molecular data using parsimony criterion, and basically we end up with massive polytomies where we have so many equally parsimonious trees that it's impossible to distinguish amongst them. And so basically the analysis tells us nothing. And then we also always have to be aware of this possibility of long branch attraction in doing a parsimony analysis. Maximum likelihood attempts to take care of a lot of this. And basically what you're trying to do is to maximize the probability of observing the data you have given that cladogram. There are multiple models of DNA evolution to use, and the recommendation is to use either AIC or BIC to choose the best model, to compare the likelihoods using those criteria. Select the one that works best and stick with that one for the analysis. So what we're going to do next time is look at the newest, the latest and greatest, and the most computationally intensive technique, which is Bayesian analysis, which makes us, at least us old-timers who were taught statistics one way, makes us look at statistics from the other side of the coin, if you will. And then also get a little bit into ways to estimate the confidence that we can place in the trees that we get. If we can't look at every tree and make absolutely sure that we've got the shortest or the maximum, the likelihood at its absolute maximum, then how confident can we be of what we're seeing is real, as opposed to just random noise in the tree? So we'll get into that next week. So take care, stay healthy, and stay safe.